Hello everybody and welcome to our module on spermatogenesis and oogenesis. Spermatogenesis is the process of development of haploid gamete cells. Haploid cells contain only one copy of each chromosome instead of two. This is unique to male and female sex cells, including sperm and oocytes. Let's start by reviewing a little embryology. The primordial germ cells are the common origins of spermatozoa in males and oocytes in females. They are derived from epiblast cells. The epiblast is one of the two layers of the bilaminar disc in the embryo. And these primordial germ cells migrate to reside among endoderm cells of the yolk sac, where they form a structure at about 8 weeks after fertilization called the genital ridges. These two genital ridges will go on to develop the gonads in the male and female. Now, even though male children are born with germ cells, the process of spermatogenesis, by which spermatozoa are generated, does not begin until puberty. Male children are born with structures in their testes called sex cords, but initially they do not have a lumen. At puberty, they develop a lumen and they become the seminiferous tubules, and this is the site at which spermatogenesis occurs. Cells going through spermatogenesis undergo a special type of cell division called meiosis, which I talk about in a separate video in the genetics section. In meiosis, diploid cells, which are 2N and have two copies of all the chromosomes in the body, give rise to haploid cells, which are 1N and have a single copy of each of the chromosomes in the body. These cells are called gametes, and this type of cell division, meiosis, is unique to germ cells like spermatocytes and oocytes. Now, in order to understand the names of all the different cells involved in spermatogenesis and oogenesis, you have to understand the chromosome content of cells going through meiosis, because cells are named according to their chromosome content. So let's review that now to make this very clear. So let's imagine that we have a cell going through spermatogenesis and it has a single copy of every chromosome in the body. And just to make this clear, let's zoom in on one chromosome, for example, chromosome 6. So let's imagine we've got a cell and it's got a single copy of chromosome 6. We would say that this cell is 1N because it has a single copy of chromosome 6. And it does not have two sister chromatids, so therefore we would call it 1C as well. Now, if we had another cell going through spermatogenesis and it had a single copy of each chromosome, we could again zoom in on chromosome 6, but now let's imagine that this cell has two sister chromatids linked by a centromere. So this cell has gone through DNA synthesis and it has two sister chromatids linked by a centromere. We would still call this cell 1N because we count chromosomes by the number of centromeres, but we would say that this cell now is 2C because it has two sister chromatids for each chromosome. So if you understand that, then you could easily imagine that we could have other cells that, for example, had two copies of chromosome 6, one that originally came from the mother, one from the father, and had two chromatids for each of those two copies. We would then call that cell 2N because it's got two centromeres, two different copies of chromosome 6, but we would call it 4C because we've got four sister chromatids. So you need to understand this nomenclature, how we decide if cells are 1N or 2N, and how we decide if they are 1C, 2C, or 4C, in order to understand the names of all the cells going through spermatogenesis. So here's a picture just to make this even more clear. So let's imagine that we start with a cell that is 2N, 2C. It has two copies of each chromosome, and I've shown inside the cell a blue line and a red line to represent any particular chromosome. Let's just say it's chromosome 6 for argument's sake. So we've got two copies of chromosome 6, and each chromosome has only one chromatid. So this is a 2N, 2C cell. The blue chromosome comes from the father, so it's the paternal chromosome, and the red chromosome comes from the mother, so that's the maternal chromosome. So as cells go through meiosis, for example, sperm cells are going through meiosis, the first thing they will do is undergo DNA synthesis, and we will then generate a cell that is 2N4C. It still has only two chromosomes, one's blue and one's red, but each chromosome has two copies, in other words, two chromatids, so it's now 2N4C. The cells will then go through the first stage of meiosis and separate into two daughter cells, which will each be 1N2C, and then the cells will go through meiosis 2 and become haploid cells that are 1N1C. So the reason I went through all that detail about chromosome content of cells is because if you understand that, then it's very easy to understand the names of all the different cell types involved in spermatogenesis. So spermatogenesis begins with spermatogonia. These cells are 2N2C. They are derived from primordial germ cells, and they are the precursor of what will ultimately become the spermatozoa. Basically, these cells are just like any cell in the human body. They have two copies of each chromosome and a single chromatid. It's shown here at the top of the drawing. Now the spermatogonium will undergo mitosis and generate two daughter cells. One of those daughter cells will stay as a spermatogonium, and in this way spermatogonia are never lost. But the second daughter cell will become a primary spermatocyte. Primary spermatocytes are the result of mitosis of spermatogonia, and they are diploid just like spermatogonia. The only difference between primary spermatocytes and spermatogonia is that primary spermatocytes have copied their DNA, so they are 2n4c. For each chromosome, there are two sister chromatids attached by a centromere in a spermatocyte. That's what makes it different from spermatogonia. DNA synthesis has been complete, and these are the cells that are about to go through meiosis 1. Once the primary spermatocytes go through meiosis 1, they now become haploid. This is shown over here at this point in the drawing. They now become what's called a secondary spermatocyte, which is a haploid cell. These cells have completed meiosis 1, they're starting meiosis 2, and they are now 1 and 2c. And then finally, those secondary spermatocytes will go through meiosis 2 to become what are known as spermatids. These are also haploid cells, but they are now 1 and 1c. And if we go back to this drawing here showing meiosis, this basically shows all the different cells of spermatogenesis. So the 2 and 2c cells are the spermatogonia. After DNA synthesis, they become primary spermatocytes, which are still diploid. After meiosis 1, they become secondary spermatocytes, and then after meiosis 2, they become spermatids, which are 1 and 1c. Now, just because we've generated 1 and 1c spermatids, this doesn't mean the process is complete. Those spermatids have to undergo a process called spermiogenesis. This is different from spermatogenesis, even though the two names sound alike. In spermiogenesis, the spermatids form spermatozoa, or sperm. The singular is spermatozoan. And these are the cells that have the capability of fertilizing an egg. So spermiogenesis is the process of formation of spermatozoa from a spermatotid. This involves formation of a structure called an acrosome. This is a cap of the sperm, and it contains special enzymes that assist with fertilization of an egg. This process also involves condensation of the nucleus, formation of a neck and tail, as shown in this drawing here, and shedding of most of the cytoplasm. So after the DNA has been replicated and the cell becomes haploid, it's not yet ready to fertilize an egg. It has to undergo the process of spermiogenesis and become a spermatozoa, and then it is ready to fertilize the egg. This whole process of spermatogenesis and spermiogenesis would not occur without the Sertoli cells. They line the walls of the seminiferous tubules and they support and nourish developing spermatozoa. They also regulate spermatogenesis. They are stimulated by FSH, and in this manner, the pituitary gland has some control over how much sperm are generated by the testes. Now, in addition to being controlled by FSH from the pituitary, the Sertoli cells are supported by Leydig cells, which release testosterone. The Leydig cells are very close to the Sertoli cells, so they release testosterone, which acts as a paracrine, meaning a hormone that acts on a nearby cell without traveling through the bloodstream. And the Leydig cells, as you may know, are stimulated by luteinizing hormone. So what this means is you need both follicle-stimulating hormone and luteinizing hormone for normal spermatogenesis to occur in the male. The Sertoli cells also form something called the blood-testis barrier. There are tight junctions between adjacent Sertoli cells. So if you look at this drawing on the screen here, this red structure with the number 7 is a Sertoli cell, and you can see that it has these tight junctions here. So behind the tight junctions, you find the spermatogonia. Remember, those diploid cells that will ultimately undergo spermatogenesis. On the other side of those tight junctions, you can find developing sperm. You can find secondary and primary spermatocytes and spermatids and spermatozoa. So the apical side, which is the side towards the tubule, is where meiosis and spermiogenesis are occurring. That's on this side of the tight junction. The basal side is where you find the spermatogonia undergoing cell division. So these tight junctions and the blood-testis barrier formed by the Sertoli cells isolate sperm, they protect them from autoimmune attack, and they also allow the Sertoli cells to control the environment and drive spermatogenesis. So just to review, because these are important points, listed on this slide are the cells you can find in the seminiferous tubules other than the spermatozoa and the spermatocytes. So you can find spermatogonia, which are germ cells. They're behind the blood-testis barrier. They're separated from the tubule by Sertoli cells. You can also find the Sertoli cells, which line the tubules and support and regulate spermatogenesis and form that blood-testis barrier. And then finally, you can find the Leydig cells. They are outside of the seminiferous tubules. They're in the interstitium between tubules, and they secrete testosterone, which acts as a paracrine to stimulate the Sertoli cells. And finally, here's a picture of a seminiferous tubule like you can find in your textbooks. The lumen of the tubule is in the middle. You can find the Sertoli cells lining the outside here. You can find the Leydig cells outside of the tubule. They release testosterone that goes across the membrane to stimulate the Sertoli cells. Spermatogonia are also along the outside. Remember, they are held behind those tight junctions of the Sertoli cells. And then you can find primary spermatocytes and other spermatocytes and spermatotids towards the center, where they are in the process of generating spermatozoa. Now let's talk about the process of oogenesis that occurs in females. So in females, the primordial germ cells form oogonia, which are 2N2C, just like the spermatogonia in males are 2N2C. But unlike in males, where spermatogenesis doesn't begin until puberty, oogenesis begins in utero. Those oogonia begin to divide in utero and form primary oocytes, which are 2N4C, just like the primary spermatocytes were 2N4C. And in fact, the female will form the maximum number of primary oocytes that she will ever have in her life by about the 5th month in utero. This is about 7 million primary oocytes. After this, oogonia and oocytes will begin to die, and she will never have any more oocytes for the rest of her life. The oocytes will become surrounded by cells, and they will form the primordial follicles, which you may know can go on to generate an egg during the menstrual cycle. Now those primary oocytes, those diploid cells formed in utero that are 2N4C, will begin meiosis I, but they will arrest in prophase of meiosis I and stay there until puberty in the ovaries. At puberty, when the female begins the menstrual cycle, a few primary oocytes will complete meiosis I each cycle. Some will form what are known as polar bodies and they will degenerate, but some will form secondary oocytes. So secondary oocytes are 1N2C, just like in the male where the secondary spermatocytes were 1N2C. These cells will now begin meiosis II, but they will arrest in metaphase. If no fertilization occurs, the secondary oocyte will degenerate. If fertilization does occur, however, the oocyte will then complete meiosis II, and the egg will become 1N1C, which has now been fertilized by a 1N1C spermatozoa. And that concludes our video on spermatogenesis and oogenesis.